so we can get started. Good morning, everyone. Good afternoon, good evening, wherever you might be. So I would like to welcome everyone to the Theoretical and Computational Biophysics group seminar today. So it's my great pleasure to host and introduce our speaker, Professor Kresten Lindorff Larsen from Copenhagen University. So by way of introduction, Kresten did his initial training in biochemistry in Copenhagen, and then he moved to the famous lab of late Kresten in Cambridge, England, to do his PhD in biochemistry. And after having a little detour into industry, actually, so he came back to University of Copenhagen as a postdoctoral fellow, as a research fellow, EMBO research fellow, and then he joined the faculty there in 2005 as an assistant professor, Department of Biochemistry. But then he decides to come to this side of the Atlantic Ocean and work for D.E. Shaw Research Group for about 4 years, where he participated actually in groundbreaking work that many of us know about, on protein folding, very long simulations and beautiful analysis, really connecting what we know in biochemistry to what we can achieve in simulations very nicely. But then in 2011, he goes back to University of Copenhagen again, this time as an associate professor. And since 2016, he has been a full professor in the structural biology and NMR, and the Center of Protein Science at the Department of Biology, University of Copenhagen. So he is actually, he has some major grants, multi-million euro grants actually there. He has been directing a PRISM Center there, which is funded by Novo Nordisk, and also a BrainStruct Center, which is funded by Lundbeck Foundation Initiative, among other things. He has received a co-recipient of a Gordon Bell Prize a few years ago when they were working on the protein folding problem pointed out. And he's also heading the biocomputing core facility in Copenhagen. Research was, many of you know Kresten, I don't need to spend too much time here, but he is combining techniques of biochemistry and biophysics with the structural biology, bringing computing together with these areas to address many different areas, actually, including some of the challenging areas that we are facing, namely intrinsically disordered proteins, for which essentially we don't know any structure and how to handle them. He has been also, as I pointed out, behind some of the longest simulations that we have in the literature, first millisecond simulation of proteins, for example, and he has been involved in actually more accurate force fields that we need for these long-term simulations. So he has been cited almost 18,000 times, and he has a Google Scholar H index of about 50 as we speak. Well, thank you very much, Kresten, to be here at your evening hours, and we look forward to your seminar. Please.
Thank you very much for that introduction. And in particular, thanks everybody for coming. And today I'll talk about, one of the two major research directions here in the lab of sort of at the interface between molecular simulations and various types of biophysical experiments, and how we sort of trying to, both develop and apply methods sort of to bridge the two worlds. And so I'll talk about, roughly two different things that one can do in this area. One of them is something we've been working on for quite a few years, which is sort of trying to refine conformational ensembles using experiments. And I will focus on some, let's say, not too complicated applications, things that are published, but I'm happy to discuss more complicated things that we are working on, maybe afterwards. And then I'll talk about, in the context of disorder proteins in particular, how we are trying to improve coarse-grained modeling using the fact that simulations and experiments don't always agree, and how we can sort of maybe use that as a starting point for refining models. So I probably don't need to give much of an introduction at the interface between experiments and simulations, but nonetheless, I'll just sort of preface it with the fact that we sort of conceptually think about working in two different ways. One of them is that we have some sort of system that we run simulations of, and as many other scientists, we like to have coffee while our simulations are running. And then once we run our simulations, we want to figure out whether they are meaningful or not. And so we often try to make predictions about observables, and then we ask our collaborators, or we go into literature to try to find experiments that we can sort of compare with. And of course, this is one of the many ways that we and many other people have sort of been benchmarking simulations, both in terms of sampling, but also in terms of force fields. So this is what we call forward modeling. And then what we also often do is that we are contacted by people who do experiments, or we have collaborations with people who do experiments that measure some set of observables. And then, this would be then the starting point. And then we say, okay, if we want to understand what these experimental data mean, maybe we can use simulations as an interpretation tool rather than a prediction tool. And so we try to construct, specifically conformational ensembles, or sometimes even kinetic models, to try to explain the data and sort of use it as a sort of fitting tool for interpreting the data. And I'll also talk about that. This is sort of the kind of inverse modeling, and this is what I'll mostly talk about, at least initially. And so these two worlds, or these two different ways, they're sort of they're in some sense disconnected, but also not unrelated. And in particular, all the work that that the community has been doing in terms of both improving sampling but also improving force fields is making our computational models as a whole much better. And so that means that even in the cases where we do have some data, we can sort of get out more information from less data. And sort of this is illustrated with this kind of continuum, where over here on the right hand side we would have what we would traditionally call maybe the protein structure determination, where we have probably a relatively simple computational model, a force field that maybe only has excluded volume and fixed bond lengths, but then high resolution information, and we just you do very simple simulated annealing, and you get out nice structures of proteins, but driven very much by the experimental data. And over here on the left hand side, you have some sort of fancy computational model. This could be a very accurate force field, or some other high accuracy model where you just plug in a sequence, and that's really all you need. But of course, as we are getting better and better force fields or energy functions, better sampling algorithms, we can sort of often end up in this kind of middle regime where they're not really good enough on their own to make really good predictions, but with a little bit of experimental data, this could be sort of low resolution data in the terms of maybe a few distance restraints or some X-ray or neutron scattering, etc. We can still say something meaningful about these ensembles by sort of integrating the two worlds. And so as we as a field are moving towards this left in terms of force fields and sampling, we can either tackle more and more complicated systems, or we can tackle sort of fixed complexity systems with less and less data. And so those sort of the improvement of force fields and the integrative modeling in many ways go very much hand in hand. And so I'm assuming that at least those of you who've done computation have been in a situation where you run some set of simulations and you have also some set of experimental data, and you try to compare the two, and they don't agree perfectly. And perfectly, that of course is a very can be a quite a qualitative kind of problem. If you have good error bars, if you know that your simulations are converged, you can compare with sort of more statistical models, and then we are often faced with sort of a decision on how we move forward. And one completely valid approach is say, this is the data, this is the simulations, we're trying to interpret it, we'll publish it, and we'll move on. Another approach, which I'll talk about, is how one can sort of bridge the two to refine the ensemble specifically using experimental data. So this would be sort of a very local system-specific refinement using the data. And this is what I'll start to talk about. And then what we can also do, and of course, this is not a new idea, is that we can also try to collect a little bit more data and then sort of say, well, maybe we can try to tackle the underlying problem a little bit more in depth. And so we can sort of try to maybe improve the actual force field that we're using the deviation to the experiments as sort of as a target function. And of course, we have to be careful, in all of these cases about overfitting the data. And just a reminder, this should not be particularly necessary to everybody here, but we should just always remember that nearly all experiments, all experiments that are not effectively single molecule experiments are ensemble averages. And so this is something that's very important to keep in mind when we interpret the data. And I like to illustrate this with this, very simple and a little bit, silly example of attaching a little vector to your coin. And so if someone, measures the average spin of a coin, and maybe it's averaged over long, many flips and Avogadro numbers of coins, then assuming that the coin is sort of unbiased, right, you would get 50% spin up and 50% spin down, and so the average vector, that you would measure would be a zero vector. And so if I have my experimental colleagues that are flipping coins, and they come to me and say, Kresten, I measured the average vector of a coin and it's a zero vector. Can you tell me the structure of the coin? What I would do is I would put my coin in my computer and I would sort of sample orientations. And if I was, forgetting this ensemble averaging, I would end up with this kind of coin standing on its chest down the right and say, yeah, I found a solution. This solution here matches your data perfectly. So now I can tell you what the structure of the coin is. And of course, this is a very silly example, but it sort of illustrates the fact that you can end up with, in some cases, completely meaningless solutions if you forget the fact that actually what we're measuring is not the orientation of a single coin, but an average over, over many coins. And so, we have to do something about this when we're doing the modeling, and there are many solutions to this. Some of these are sort of outlined in this review that we wrote a couple of years ago that I can recommend that you read. It sort of, I think, fills a nice little niche in terms of being relatively technical without being super technical, but addresses some of these things, slightly beyond maybe the conceptual level. And there are, I'll in the next slide, I'll also show you a couple of other references to other people's work. And in principle, there are sort of at least two directions one can take with this. So the starting point would be that we have some sort of force field that is a general force field that we would use. And then we want to integrate it with this kind of data. And we can sort of go down two routes. One route is that we can sort of directly plug in the experimental data sort of to bias the force field. And then we can sort of come up with what we and other people would call sort of a biased force field that has some system-specific correction that we can then run simulations under and then get out distributions that sort of take the experimental data into account during the sampling and get out distributions over whatever observables we might be interested in. Or an alternative approach is that you can take your standard force field, you can run simulations, they will not necessarily agree with the experiments, and so you get out some probability densities, but then after the sampling, you can then reweight the ensembles in some statistical way, and there are different approaches to do this, but we sort of use the data after we've done the sampling, and we can then construct ensembles that improve agreement with data. So you can sort of either put in the data before or after the sampling, and in some cases they are equivalent in terms of the solutions, and there are pros and cons to the different approaches that we can discuss maybe later if you want. What I'll talk about is in particular our own work on doing this sort of last thing where we sort of first do the sampling and then reweight. There are many people who worked on this. There's in particular really nice work from Gerhard Homer, but all of these people have contributed. And many other people have contributed to this. In my group, this was sort of mostly spearheaded by Sandro Botaro, shown here at the bottom right. And the basic idea is, is again, quite old and comes from many other fields, and that is sort of that we say, let's say we have some sort of simulated ensemble, represented here by this red distribution over here, and we calculate some observable that we can measure. This would be sort of the average we would get. This would be this red dashed line here. This could be the average distance measured by an NMR experiment, for example. And then we compare it to the experiment, which is this black dashed line over here, so they don't agree. How can we reweight the ensemble? Well, we could, there are many ways of doing that. And then the way that we approach would be what we would call the maximum entropy, solution or the minimum cross entropy solution, where we simply make the smallest perturbation to our simulation that we treat sort of as a statistical prior. And in this case, it could, for example, mean that we sort of would decrease the population of this state, increase the population of this state, so that we move the average from, from where it is, which is wrong, closer and closer to this true, measured average over here. And again, there are many arguments why this is a good idea, and discussed in some of these reviews, and there are other ways of doing it also. So one way to think about this, and this is also sort of originally described in this paper from Gerhard Hummer, is in terms of let's say more thermodynamic kind of framework, is that we are optimizing a function here that I've called gamma, sort of by analogy to a Gibbs free energy, and it contains two terms, this chi-square term here and this entropy term over here. This chi-square term here you can think about as an enthalpy. This measures the deviation to the experiment, so we want to minimize the chi-square to sort of fit the data, but at the same time we want not to affect this entropy, which measures the deviation from the prior, and so we want sort of to at the same time try to maximize this entropy over here, and the balance between this enthalpy-like term and this entropy term is set by this temperature-like Lagrange multiplier theta here. And so this is very similar sort of to how we would think about a free energy kind of system, although what we're doing here is that we're optimizing these weights that are then the weights of each individual frame in our MD simulation. So we'll tweak those weights to minimize this chi-square, but using this entropy, this cross entropy to the starting point as a sort of regularizer, so to avoid overfitting. And so we are very explicit about this theta parameter here, which is sort of a fudge parameter that really acts to sort of regularize the problem, and we need to choose it in a way that I'll describe to you in a second. So here's a 2D toy example just to illustrate this idea. Over here on the left is a sort of two-dimensional energy landscape, and the marginals are shown over here are called X and Y. And this black and gray histograms would be sort of nature's force field, the one that we would like to know, but that we don't have. So this is sort of the truth that we don't have. And we can then generate sort of this two-dimensional energy landscape here. And this would be sort of the average of X and Y under this energy landscape. And some realization of an experiment might be this purple point over here. So let's imagine that we can get the average of X and Y, and that's all we know. And then we have some force field that is not perfect. This would be this red force field over here that we can sample. It's not perfect. You can see the marginals don't really agree with this. So when we sample the red force field, we get this average that down here. So we have an average that doesn't agree with our experiment. And so what we're doing is that we are sort of effectively reweighting then the samples that we get from the red force field, so to move the average closer and closer to the purple point. So this is illustrated over here on this gray line over here. And as we are walking down this gray line, we are changing this theta parameter that's changing the balance between fitting the force field that is sort of fitting the data that's illustrated here with this chi-square on the y-axis versus this phi effective on the x-axis, which is another way of expressing this entropy. So it's the exponential of the entropy, which effectively just measures what fraction of the original MD frames are sort of retained in this reweighting procedure. So if the temperature is very high, we are just using the prior, we have a poor agreement with the experiment. As we're decreasing the temperature, we're fitting the data more and more closely. We're sort of decreasing the chi-square, but at the cost of having to sort of discard or downweigh some of the frames in the MD simulation. And so what we do is that we sort of try to come up with a balance of trying to say, well, it's okay to discard a little bit of the MD to sort of fit the data, but at some point you really need to discard a lot of the data to really fit it even a little bit better. And if we trusted all the error bars and we've charted all the statistics perfectly, we would typically go for something like a reduced chi-square of 1. But unfortunately, in many, many cases, it's really difficult to actually get good error estimates of many different things, including the error that comes from the sampling, but also the error that comes from the experiment, the error that comes from the way that we calculate the experimental data. We often don't have good control over those. So instead of just going for a specific target chi-square, we sort of try to find some kink in these kind of curves here, and then we try to explore what happens if we choose this theta-like parameter a little bit higher or a little bit lower. And what we also, and what you will probably many of you realize, that we are sort of doing a sort of a kind of free energy perturbation here. And of course, the less and less overlap you have, right? This corresponds to decreasing this phi effective a lot, the more noisy it gets. So when this phi effective gets very small, it's also really an indication that there's little overlap between the force field that that we have available and whatever nature's force field is. And that's also a sign of warning that we shouldn't try to do this kind of free energy calculation too accurately. But if we do this, we pick some sort of point here in the middle, you can get something like this down here. You can see we've essentially moved this to sort of close to the experimental value. You can now see the blue distribution here is the reweighted distribution. You can see it's a little bit noisy over here because we essentially started with something that had very low populations, and that's why we haven't pushed it too much. But you can now see the distributions, the blue distributions are now much closer to this nature's force field that we didn't actually have access to. We just had, two measurements, X and Y averages, but you can still see we can say something more meaningful about the distribution of, samples that we would get out. And, we can discuss the philosophy about this later if you wanted to. Here's just, a single slide with one example illustrating this with a biomolecular example. This is alpha-synuclein. This is sort of combined work from a couple of different papers, mostly by Mustafa Karab, and then another paper from Francesco Pesce, and then nice new small angle X-ray scattering data that Line measured. And so over here are 3 ensembles generated either by 2 nice long empty simulations from the short research, in particular from Paul Robustelli and Stefano Piana, with 2 different force fields, and then also flexible Meccano, which is like a very simple mod for generating unfolded states. And you can see they sort of all give quite nice and expanded structures there. Those 2 force fields are good force fields for simulating disordered processes. They give slightly different distributions of the radius of gyration. And if you calculate then the small angle X-ray scattering data, you don't get perfect agreement with the data, but you get pretty good agreement with the data. So you would get something that would look something like this red line up here, comparing to the gray line here, which is this experimental small angle X-ray scattering data. And then we can do this kind of reweighting where we essentially change the weight of the different frames to fit the data. And so you end up with something like this blue line up here. This is for one of the force fields. And you can see, so we can fit the small angle X-ray scattering data. And now you can see over here on the right-hand side what happens, for example, again, if we look at the radius of gyration, and you can see you get up, 3 different ensembles that are different from the initial ensembles. But one thing that's nice to see is that they're all much closer to each other in terms of the radius of gyration. So they all become sort of more, similar to each other because we've now fitted them all to the same kind of experimental data. And this is something that's true in general, is that, if you take different force fields or different flavors of sampling and you sort of fit it to some common set of data, they all become, more or less homogeneous. And what I'm not showing you, but is described in some of these papers, is that if you picked, some really poor force fields, you would not be able to get something that looks like this. So then, this kind of free energy calculation would just be too noisy, or you wouldn't be able to fit the data. So your statistical prior needs to be not awful for this approach to work, because otherwise you really have to discard essentially all the frames in the MD simulation. So a slightly more complicated example I just wanted to show you also because there was, has been so much nice work done on these systems at your end. This is some work we did a couple of years ago, in particular simulation work done by Tone Bengtsen when she was in the group, and together with Lisa Arlett's group that did the scattering data. So this is studying a slightly more complicated system, in particular these small nanodiscs that many people like to use to study membrane proteins in sort of solutions. And the starting point for this was actually sort of an interesting observation, which is that there was a really nice NMR study on the protein conformation of nanodiscs, of one of these small nanodiscs. And this showed that these nanodiscs were essentially, round if you look at them by NMR. So the NMR structure is sort of, at least on a simple level, sort of a nice circle. And this fits, both residual dipolar couplings and NOEs measured by NMR. These are filled with lipids, but the NMR didn't look at the lipids. And then at the same time, Lisa's group had looked at earlier, and we also had some new data in this new paper, looked at them by neutron and X-ray scattering. And if you sort of try to fit a simple geometric model that describes these nanodiscs, and you want to sort of come up with a single, geometrical model that fits the data, you get something that has to be, elliptical. So a circular kind of conformation does not fit the data. And so you have this kind of apparent conflict between the NMR data that suggests sort of a round shape and the scattering data that suggests like an elliptical shape. But of course, in both cases, they're both sort of, single conformal representations of something that's an ensemble average. And of course, if the ensemble average is extensive, then, with this simple coin example, for these kind of things can be misleading. And so what we and Tone did was that we ran, a bunch of MD simulations starting from the NMR structure and sampled, essentially some ensemble of this here, some clusters up here. These are the blue sort of are the cluster centroids, and the red lines here are sort of the NMR structure. And some of them are, elliptical, in one dimension. Some of them are elliptical in another dimension. Some of them are sort of roundish. And then what Tone did, actually then compared these simulations both to the NOE data, but also to the small angle X-ray scattering data. So this is shown over here on the right, where you have the experimental data in red, and you can see here that the MD ensemble gets this kind of minimum correct, but there are some discrepancies over here. And if we calculate the scattering curves from just the NMR structure that we then plug in some lipids in, you can see that the NMR structure sort of misses this kind of minimum down here, but gets this kind of shoulder shape over here in this intermediate Q regime. So the MD gets some aspects right. The NMR structure, this sort of round things, get other things right. And so what Tone then did is that she applied this kind of machinery that I talked about before, this kind of reweighting machinery. And if I can get my slide to change, here we go. Then you can see that, she can fit the data, right? So we're fitting the data, so we're not really predicting it anymore. We're using the simulation as an interpretation tool. And so she can construct ensembles that, now nicely fit both this kind of regime over here and this regime over here. And, we're not fitting the clusters, but I'm just representing the data during clusters. So there is essentially some weights of the different clusters that, before the reweighting, this was like 50%. After reweighting, this is about 30%. This one was upweighted a little bit from 30 to 50%, and this one is about 20%. And if we then characterize each of the structures in these ensembles in terms of how round they are, and this is sort of this number square root of C is effectively the deviation between the two major components of a sort of gyration tensor. So essentially mentioned how not round is the structure. You can see that there's some distribution over here of sort of acylindrical shapes, that is sort of, some structures that is about as round as the NMR structure, some that are about as elliptical as this average, Sachs structure, but sort of something in between here. So by having sort of a distribution of ellipticities, we can fit all the data, and they fit both the NMR data and the small X-ray scattering data. And so when you have sort of an elliptical shape that's sort of fluctuating along the two different axes, if you look at it on average with scattering, it looks sort of on average elliptical. And if you look at it sort of on average with NMR, it looks sort of round on average once you average the data. And so, this is just one example of how we can use, MD simulations that, don't actually fit the data perfectly, but we can still use it as an interpretation tool to try to say something, in this case, about, shape fluctuations of nanodisk. And of course, this would be the starting point then for, putting actually membrane proteins in this. And these are things that we've looked at later. So this tool that we call Bayesian maximum entropy, and we have some code that's called BME that is available on our website. You can download and try. And we've made it hopefully relatively easy to use, and we've used it in a bunch of different systems. I'm showing you, some subset of this here, not to say much else, that we've tried to make it relatively versatile and easy to use. It's not perfect, and there are some things that you need to be aware of in terms of how well you need to be able to sample, and all these things that are discussed in some of these papers, and we can discuss them later. But I think it's a relatively simple to apply tool for interpreting data, and in contrast to other methods that use the data to bias sampling, which I think in principle is often a more powerful approach. This is much easier because you can really run all your MD as you normally do. You can use enhanced sampling, you can use metadynamics, then you just need to update your prior with those weights. And then whatever kind of fancy sampling algorithm you might have, you can use it, and then you can plug in the data afterwards. And that makes many things a lot more convenient to do. And so I'm illustrating here now two of these systems that we looked at with this, but I won't actually talk about what we did in terms of this reweighting, but this is sort of a segue into the next kind of problem that we ended up addressing. But we sort of ended up doing two projects that were sort of related. One of them, was a work done by Andreas Larsen, again together with Lisa, on these 3-domain protein tier 1, where we had nice X-ray and neutron scattering data. And over here on the right is data from Tanya Mitra's group in a collaboration with Tanya Mitra's group, in particular small angle X-ray scattering data measured by Martin, where Emil Thomasen from my group did a bunch of simulations of this, two-domain protein H9P1, sometimes has this purification tag and this long disordered tail. So we were running coarse-grained simulations of this with Martini. And one of the things that we found in both cases, this was with a preliminary version of Martini 3, but this also holds true for the final version of Martini 3, is that when we compared to the actual scattering data, we actually found that the ensembles that were generated by MD were quite a bit too compact compared to what the experiments did. They were about 25% compared to the experimental ranges of gyration. And so in these papers, what we did is we came up with some ad hoc modifications that you can read about in the papers. And then we used this kind of reweighting against the SACS data, trying to explain what was going on in terms of the conformational ensembles. And this is described in these papers, and I won't really talk about it. But instead, what I'll talk about is some more recent work we've done, trying to sort of get a little bit less ad hoc in the kind of modifications that we've been doing to Martini. So what we've been doing is essentially we say, okay, if Martini simulations of disordered proteins and multi-domain proteins give rise to structures that are a little bit too compact, maybe we can come up with some more general correction to the force field that at least enables us to look at these kind of protein systems, without having the data in hand. And what I'll talk about in the next couple of slides isn't perfect, but it's essentially always makes things slightly better or a lot better than the plain Martini, at least, and this is an important point, at least when we look at protein-based systems only. And this is particular work again by Emil Thomasen, but with contributions also from Francesco and Mette and Giulio. And so what we did is we collected data here for a bunch of disordered proteins where we had nice small angle X-ray scattering data, and we ran relatively long Martini simulations of these. And if you just calculate the ranges of gyration from Martini for these disordered proteins, you can see we get a nice correlation between the experimental and the calculated range of gyration, but with a slope that's less than 1, meaning that the structures are all a little bit too compact. And so what we then did was, inspired by first the work that we'd done earlier for the two tier 1 and H9P1 system, but also work that Rob Best and others have done for all atom proteins, we try to tinker a little bit with the force field, and in particular say, if the structures are a little bit too compact, maybe by strengthening the interactions between protein and water, we can make the chain swell. And so we sort of introduced this parameter lambda in the Martini force field that effectively scales the interaction strength between protein and water. And so this is what I showed you before. This is plain Martini, lambda equal 1. And then, for example, this is what you get if you increase the protein water strength by 6 or 8%. And now you can see we get an equally good correlation, but now with a slope that's about 1. We can also, instead of comparing to the RG, which isn't really an experimental measurement, we can compare directly to the SACS data. And you can see down here again that the chi-square, when you compare to the SACS data, for each of these lines is a different protein, we can see that we get improved agreement with the SACS data, and this is sort of the average chi-square. And you can see again we get sort of a minimum sort of around here about 8-ish%. What we also did is we ran the same kind of simulation for these multi-domain proteins. I'm showing you data here for 3 proteins, and you can see again that we get improvement as we increase lambda, but it looks like at least that the optimal rescaling is somewhat different for these kind of multi-domain proteins compared to the purely disordered proteins. So we have a correction, and choosing 1.04 or 6 or 8 improves things relative to not rescaling things. Note that this is a log scale, so these can be relatively big changes. There's not a single universal thing that we've just fixed everything, right? So you can either take this and you can pick some value in here and be pretty sure that it's better than doing nothing, or you can try to sort of have some experimental data available, then you can maybe do this slightly better. And of course, we're trying to figure out maybe something that's a little bit more physical, sort of we can come up with something that's a little bit more universal. But I'd say this is a nice step along the way. So they are improved, but as I said, imperfect. But for example, if we take alpha-synuclein and we run simulation of 2 alpha-synuclein chains under conditions where this thing should be essentially monomeric. If we just take alpha-synuclein with plain Martini, we essentially see it dimerize almost 80% of the time. If we run this protein FUS under these conditions where it should also be essentially monomeric, if you run it, on the plain Martini, I'm not showing it here, it dimerizes, 100% so much that we can't really get an error estimate of how much it dimerizes. So I'm not showing it here, but you can see that, with this increased thing here, at least, we can start to see association and dissociation under conditions where these things. But then again, and this is described more in the paper, we've also run simulations of dimerization of free ubiquitin. And, the answer is it's a little bit complicated, and we don't really have a full understanding of what's going on, but it looks like that the rescaling necessary for folded proteins is somewhat different from the rescaling necessary for disordered proteins. And so we're trying to figure out, what's the best sort of approach to encompass both things. But if you're working on pure protein systems, a little bit of rescaling will generally help, either a little bit or a lot. But realizing that this, picking a single parameter out of a complicated force field and tweaking that is somewhat arbitrary. Now I'll talk about here at the end some work we've been doing on trying to, address, the whole force field simultaneously, or almost all the force field, by targeting experimental data. And so this is work that was just published. This is work by Giulio Tosei, and actually builds upon some work we did more than 10 years ago by a master's student, Anders Nørgaard, and there are other people who've done work along these kind of lines. And so the idea was, can we more systematically parameterize the coarse grain model using experimental data? And so in this particular case, what we've done is that we've started with sort of a C-alpha based model. We started with some parameters that came from a mod that was developed by G.T. Mittal and Rob Best and colleagues. It's called the HPS model. The parameters came from some other work and that they used extensively to study IDPs, but also in particular liquid phase separation. And then what Giulio, together with Tia and Lamont, did was that essentially we re-optimized this model using experimental data only on isolated disordered proteins. So we essentially parameterized a new force field targeting the experimental data on monomeric proteins, and then we tested it and applied it on dimerization, and I'll talk about, and also calculating assemblies of disorder things. Because, and this is not our work, but many people have shown, both for homopolymers, but also more lately for heteropolymers, that there is sort of a correspondence between the driving forces for forming, condensates and the interactions within single chains, which of course, makes intuitive sense, but also, as others have shown, also holds true quantitatively. And so again, this is the kind of model that we're using. This is again this HPS model, which is sort of our starting point, and it sort of has a parameter that in this model is called a sort of a hydrophobicity kind of parameter. Although maybe we should just consider the stickiness parameter. And in this particular implementation, right, the least sticky residue is arginine, and the most sticky residues are phenylalanine, proline, isoleucine, sort of the more hydrophobic residues. And so the interaction strength between two amino acids is just a combination of the the stickiness of each of the parameters. And again, this is work from from G-Tain and Rob and others. But instead of sort of picking just a single hydrophobicity scale off the shelf, we said maybe actually we can use a bunch of different hydrophobicity scales, and we'll use them as sort of a statistical prior for our optimization. And so what Giulio did is he collected a bunch of different hydrophobicity scales and essentially constructed a distribution of hydrophobicity scales. This is illustrated with this color bar over here in the background. So arginine, again has a high density down here. This is pretty, unsticky or unhydrophobic. And other things, have sort of a more broad distribution. We see proline has a relatively broad distribution, but you can see again that there is some spread on how different scales talk about the hydrophobicity. So we won't really use these distributions in our force field, but we'll use it as a prior in what I'll talk about in a second, which is a Bayesian optimization approach of the force field. And again, I'm showing you on top of here this HPS scale. This orange line is just the average of our prior, which we also use as a starting point. So again, there's no specific reason why an average over these scales would make sense, but it's just one other way of picking a hydrophobicity scale. And then there's this green line here, which is another scale that in particular G.Tain and Mittal also have used in these kind of things. This is called the Urey scale, and you can see they all differ. And if you look at the literature of hydrophobicity scales, there are many hydrophobicity scales, and they are all different from each other, or many of them are quite different from each other. And so before we do anything, let's just characterize sort of a couple of these scales against some experimental data. Blue is again this HPS scale. orange is this average scale of the other ones. And we took, a bunch of different proteins for which we have, in this case, small angle X-ray scattering data. And so over here is data on what we sort of call hydrophilic. These are relatively expanded proteins. And you can see if we run MD simulation with this average scale, actually they give pretty good predictions of the radius of gyration. So the deviation is close to 0. Whereas if you use this HPS scale or this particular implementation of the HPS scale, they're all way too compact. On the other hand, if we take variance of the liquid low complexity domain of A1, which is sort of more hydrophobic, you can see that this HPS scale actually predicts those radii gyration pretty well, whereas this less sticky scale over here actually overestimates the radius of gyration. So you have one energy function that works well over here and another energy function that works well over here. And so saying they're not neither are perfect, although they sort of work okay in different regimes. And so what Julio did, and again, this is based on this old work from others, is that we said, okay, can we then optimize the parameters in sort of a Bayesian self-consistent fashion, where we now in this case sort of construct a target function, a likelihood function that this is a log likelihood function, so it simply corresponds to two terms. These two terms over here sort of together measure the deviation against some sort of data. This would be radii of gyration from small angle X-ray scattering and NMR data, in particular paramagnetic relaxation enhancement data. So these two terms over here measures essentially how far away are we from data that we can predict from simulations. And then there's this prior over here. This is essentially the log of the prior. So this is this how far are we staying from within this distribution of stickiness scales that I showed you previously. And so what we do is we start with some force field parameters. These are then these stickiness parameters. We run coarse grain simulation of these. We calculate the radii of gyration and the NMR data. We compare to the experimental data, and then we sort of nudge these lambda parameters and compare to the experimental data. And we can do this without running, we can change the parameters a little bit without running new simulations, just by reweighting the existing simulations by changing the lambda parameters. And so we can effectively change the parameters, optimizing the agreement with the experiment. And then once in a while, we change the parameters too much, so we essentially are out in the tails, and then we have to resample, and we can sort of do this over and over again. So we run simulations, we reweight them against the data. Once we've reweighted them too hard, we rerun simulations with a new force field, and we essentially optimizing the force field parameters so that we get good agreement with small angle X-ray scattering data and NMR data. Again, using this distribution of hydrophobicities as a regularizer or as a prior. And this is illustrated on the next slide once that changes over here. I don't know what's going on, but hopefully, here we go. So here we started by collecting a bunch of data. So we have, quite a lot of data. These are all proteins for which we have, radii of gyration from small angle X-ray scattering data. These are proteins where we have paramagnetic relaxation enhancement data, and for some of them we have both. So quite a lot of data that we can compare to. And then I'll show you now what this optimization procedure is doing. So this is one starting point for the optimization. So this is the scales, and when the movie will play in a second, what you'll see is that we are sort of optimizing these stickiness parameters. Down here on the left, you'll see what the conformational ensembles look like, and over here on the right, I'll just show you the agreement with two sets of data. The PIE data from synuclein and some the radii of gyration from synuclein. But we are optimizing not just the agreement with these two, but the agreement for all of these data sets. But I'm not just showing you one of them, right? So we're not just targeting these two data points, but all the data points. And so essentially we can see that we're sort of optimizing all these parameters, and you can see we're sort of fitting and getting closer and closer to the PIEs and getting closer and closer to the radii of gyration. At the same time, you can see down here that the protein essentially swells because we started with parameters that made that made the chains way too compact compared to what the experimental data, both for synuclein, but all the other proteins get. And so this would then be the final parameters we get out of this optimization protocol. Again, optimizing not just on two data points, but on many, many data points. And so, this is then the agreement that we would get. We actually have three different mods. They're called mod 1, 2, and 3. These are slightly different hyperparameters in this balancing between the different data and the prior, and they are not quite identical, but they're pretty similar. You can see we get a good agreement with the radii duration across many different proteins. You can see again, this is this HPH model, this is the Hura model. This is zooming in on variance just of the A1 low complexity domain. So these are all proteins of the same length that just differ in the amino acid composition. Again, we get a pretty good correlation coefficients of 0.8 to 0.9 across these proteins, much better than the other models that we've sort of optimized it. And you can see down here again that these models that use slightly different hyperparameters, they are pretty similar but somewhat different. And again, you can see up here the previous parameters and the prior that we use. One thing that was sort of, I think, nice to see is that we can actually, the data and the prior actually defines the force field pretty well. So up here, I'm showing you two different optimizations using the same target function, but starting from two different force fields. One of them is this, average force field. The other one is just starting with all the parameters being the same. And you can see we more or less get, a well-defined force field, from two completely different optimizations, suggesting that the combination of the data and the prior really defines the force field particularly well. And there's a bunch of things that's interesting with this. One thing that's particularly interesting, I think, is that, arginine is a very sticky amino acid. This is not something that's new. This is something that people have known in the phase separation field, but that's something that falls out very nicely from this optimization, that this parameter needs to be high really to explain the data. Tyrosine is high, alanine is actually quite low, and a bunch of other things that sort of, in hindsight makes sense, but that sort of nicely falls out of this optimization. So now we've optimized it. Let's try to compare to some data that we didn't print. And we've done a bunch of different things here. I'm showing you just one examples where we, instead of just looking at a single chain, we actually took two IDPs, put them in a box together. We looked at the interaction between the two IDPs. This you can do, for example, by NMR, by spin labeling one protein and putting NMR active isotopes in the other protein, mixing them together and looking at paramagnetic relaxation enhancement between the chains. Those things we capture pretty nicely. There's a long, complicated story behind this, but that works pretty well. Over here is actually a little bit of a simpler, but I think quite actually a rigorous test, which is just, again, looking at how much the proteins are dimerizing. And in particular, we took 3 proteins where there's nice experimental data suggesting that these proteins under the concentrations and conditions we're running should just be monomeric. And you can see that they are effectively monomeric under these conditions. Whereas if you used one of these more sticky force fields, they would form much stronger dimers. Whereas these two proteins that are known to form phase separate, they form a little bit of dimer, which we also expect, although it actually turns out to be very difficult to measure experimentally how much dimer they should form. But at least, in this case, it makes a lot of sense. And this is actually, getting these things not to dimerize is actually not as trivial as it sounds. So of course, what we and other people were interested in using these models for is to look at, these kind of multi-chain systems. And so we again, inspired by other people, we ran, multi-chain simulation with, maybe 100 chains, in sort of what we call a slab geometry, where we would then get self-assembly of, chains in a condensate, but also with some, some chains in the dilute phase. And so we can run these, for long periods of time. We can calculate the concentration in the dilute phase and the condensed phase. This is the strength of these models. They're sufficiently simple that you can run them for hundreds of chains for many microseconds and get these kind of things relatively well converged. So we can then start to compare to measurements of these things. And again, there's a bunch of things that I'm not talking to you about, but effectively these are now correlations between experimental measurements of the saturation concentrations across a whole different set of proteins and also different variants of the same protein compared to the values we get from the simulation. You can see that there's a really nice correlation between the calculated and the experimental saturation concentration. And there are some details that are not with temperatures that you should look up in the paper. So it's not quite as perfect as it's looking like here, but it's pretty good. And this is something that turned out to be quite difficult to get this kind of correlation looking across very different systems. So looking at variants within a single system is a little bit easier, but looking at proteins that have completely different composition is actually quite different. And we capture this, with this kind of model pretty nicely. Although, and this is just one I want to say, that we really only optimized it by targeting, individual single chains, data on single chains, and then we're applying it now to multi-chain simulations. And so what we can now do with this kind of models, we can try, for example, to characterize what are the structural features of the single chain, the dimer chains, and the condensates. This is sort of an interaction energy map where white here means strong interaction energy. So this is the contact map of a single chain. You can see these kind of stripes. These are residues that are sticky, that are forming interactions within a chain. This is now a simulation, and again, this is of the low complexity domain of Fuss. You can see again this kind of checkered pattern. Again, now between two chains, there are certain residues that are sticking together much more strongly than other residues. And over here, these are the same kind of interactions. Now looking at the central chain in a condensate and sort of averaging over all the other chains in the condensate. So this is not symmetric, but it's almost symmetric. Whereas these are per definition symmetric, and you can see, at least conceptually, that that the pattern is pretty similar. Again, saying that there is a similarity of intra and intermolecular interactions. Again, this is not not something we've said for the first time, but again, this sort of falls nicely out of this kind of optimization. And this, of course, is why it worked, right? Because we've optimized essentially on these systems over here, and then we can sort of apply the model for these systems over here. This is, again, the same data. This for another protein. We can, for example, sum up the interaction strength, across these kind of matrices. So these are now, again, looking at the single chain, the dimers, and the condensates with these sort of average interaction minutes. You can see these kind of dips here that are then these kind of sticky residues that are then helping drive either compaction of the single chain or interactions between two chains or in the condensate. And you can see, as expected, that they're very similar, but there are also some differences. In particular, the monomers are sort of different at the end, which makes sense because, of course, in the monomer there is a local interaction that is not driving the interaction between chains here. And so in these kind of dimers, in particular in this kind of melt here, you get a more homogeneous distribution of the interactions even towards the ends. But overall, they're very similar, which of course is also why mutations, in these sticky residues here, this is again is work shown in particular very nicely by Tanya Mitak and Rohit Papu's lab, can really change compaction of the single chain that you can then really monitor very nicely as a shift in the phase diagrams of the multi-chain systems. What we can also look at, for example, is we can compare, structural properties in the dilute phase and the condensates. I'm showing you here, the radii of gyration, and all of these proteins are sort of, they like itself quite a bit, so they are sort of slightly below the theta regime, so they're slightly more compact than under theta conditions. So they have a scaling exponent that's slightly below 0.5, meaning that they like to interact with themselves, slightly better than they like to interact with water. So they're compact, which is also why these proteins in particular, helps them phase separate. And so this is the distribution of radius of gyration in both the dilute phase and or in a single chain system. But if you look then at the chain in the condensate, you can see they swell a little bit because now they're no longer surrounded by water. Well, of course, this is there's no explicit water, but in this implicit water, they're now instead surrounded by a melt of other polymers that they like to stick to. So the proteins then swell, and they swell sort of towards increasing the scaling exponent to 0.5, because now they really like to interact with their neighbors, because the neighbors are like themselves, and so they they don't really care whether they interact with themselves or whether they interact with the neighbors. So we see this kind of expansion of the chain in the condensates. So with that, I'd like to conclude and hopefully have some time for some questions. I started out by talking about how experiments and simulations go hand in hand, and if you have some nice set of data and you have some nice simulations, you can either say stop and we'll publish it as it is, or you can go down two different routes. One route, which I really we really like to do a lot, which is this kind of reweighting or biasing the simulations. This is then useful for studying some specific system that you're interested in, trying to construct ensembles that help you explain what's going on in the system. I showed you data for alpha-synuclein and these nanodisks, and there are many ways of doing this. I can recommend our own tool called BME, but there are other approaches out there. Or what we, and of course many other people have been doing for a number of years, is sort of trying to collect more of these kind of discrepancies and use that as a starting point for really improving force fields. And this is, I think, is particularly useful for these kind of coarse-grained models where there's, where doing sort of a bottom-up kind of parameterization is very difficult, because you essentially accumulate errors that really, starting with an all atom force field and sort of coarse grading it really doesn't work so well. So instead you can use these kind of iterative approaches where your target function is the experimental data. And in this case, of course, we need nice experimental data. But then, as I've showed you, right, we can really optimize an almost full coarse grain model. And of course, we would like to be able to do something similar with Martini. And of course, then we would probably need some sort of hybrid approach where we both use some more physics-based prior at the same time as targeting the data to get some consistent approach for parameterizing these force fields. So before I conclude, I'll just flag this workshop that we're organizing next year, in particular for PhD students and postdocs. You still have time to apply. If you forget the details, you can just search for my name in Greece and 2022. And this workshop on this lovely island of Spetses in May will come up, and it will be sort of an integrative structural biology with amazing speakers, wonderful location. So do apply. There's we had a first call for applications, but there's still time to apply. And with that, I'd like to thank you for your attention. I think I acknowledged the people that did the work. Tone is not here; she did the nano disk work. Emil here did a lot of the work on the Martini force field together with Francesco and Giulio did most of the work on the last bit, and Mette also contributed to the Martini work, and a bunch of other people contributed to other things along the way. And with that, thank you for your attention.
Thank you very much, Kresten, for this very nice and insightful and rich seminar. So I learned a lot myself. So I'm sure people enjoyed that. So I invite everyone to for questions. So you can raise your hands either physically if your camera is on, or you can raise it in Zoom, and then I call on you. You can ask your question. Or you can type your questions in the chat box, then we can discuss. So let me actually start myself. So this was very interesting story about the stickiness that you sort of made us aware of, that you get primarily in coarse grain simulations. So what are your thoughts if we bring in the complication of the membrane into the game? So like many of these disordered proteins, of course, do things on the surface of the membrane, and I presume you have been kind of biased toward the experimental conditions, everything in solution. But how should we handle these things when the membrane is there? So do we think actually similar kind of tuning would also work in a membrane environment? What are your thoughts, Kresten?
It's a really good question, and I don't have a good answer. And the problem is that we don't have good data. So we've used this kind of rescaling approach for membrane system. So we actually have a single a hormone receptor embedded in a nanodisk with a long disordered tail. And what we did was that we did the same thing. We rescaled the interactions between protein and water, and it worked well, but it is a little bit of an arbitrary approach. And I'll rephrase your question in a way that makes it clear what the problem is. Right. So what we did is we increased the interaction strength between protein and water. We get swelling of the chain. Right. We could have also have decreased the interaction strength between protein and protein. And for protein and water, this would have been sort of the same. But of course, if we have a ternary system, then it doesn't necessarily become the same. Right? And so we don't really. The problem is that it's really difficult to measure the interactions between these things and membranes, and so we don't really have a good handle of these things. So what we've done so far is just do the same thing, but we really need quantitative data on the interactions between membranes and disordered proteins. Sure. What I will say is that if you don't do anything, it looks like you get something that's wrong. So you get a too compact chain that then sticks to the membrane. But exactly the best approach.
Actually, all proteins come together to aggregate inside the membrane. That's what we are getting actually from our coarse grain simulation.
I mean, so you're exactly that, right? So so how we tune these interactions, I don't know. And of course, this is the downside of targeting experimental data, is you need experimental data to do the optimization. Right. And so of course we would like to understand better what the problems are at a more physical level, right? So now it's sort of, mostly phenomenological. We don't really know what the problem is. We just know that there's a problem.
Fair enough. Fair enough. Nandan, you were the first to raise your hand and type your question in the chat box. Go ahead.
Okay. So my question was, maybe I misunderstood it. So is there any way that you use bias simulations to when you fit to the experimental data? And if you use, for example, I don't know, like steered molecular dynamics, and how do you have to somehow unbias the trajectory or the data point itself before fitting to the experimental data?
Good question. So it depends a little bit on the bias and the approach. So let's say the simplest approach is if you know what the bias is, then it's easy. And so this entropy that we calculate, that's a cross entropy. So it's essentially it's P log P divided by a reference probability. And so if you have a plain MD from a Boltzmann ensemble, right, then the reference probability is just 1 over n. And if you have some sort of biased data from, whatever biased approach you have, but where you can calculate the bias, then you just use that as your reference weights in this optimization. So if you're doing something like, metadynamics or something else, then it's easy. If you have a biased simulation where you don't know what the bias is, then it also works. But then, then you just have to cross your finger and say, this is my prior. And of course, there's nothing, Bayesian statistics is pretty forgiving, right? You can define your own prior. This is your own best bet, between you and, your own, thinking about the system. And this is your starting point. And so if you say, my steered molecular dynamics is my best bet for this system, not knowing anything else, you can use that as the prior. But of course, and this is nice about this approach, the better the prior becomes, the smaller the perturbation you have to do to the prior to fit the data. Right? And so if you have a really good prior, you need to tweak it a little bit. If your prior is awful, you need to tweak it a lot. But at the fundamental level, if you have some sort of sampling approach that doesn't give, that gives a bias distribution, if you can calculate the weights per frame, with the bias or then it's easy. You just plug in the numbers and you do that. And we've done that in a bunch of cases, and that, that works perfectly fine.
So it means that there is nothing theoretically wrong to use a bias trajectory to fit into the Bayesian prior, right? So theoretically, it's fine.
It's absolutely fine. It's, if you can calculate the bias, everything is fine. Of course, it's not always easy to calculate the bias, and then it gets a little bit trickier. But if you can calculate the bias, it's easy.
Okay. Thank you.
Okay, so let me go to Jale, and then David, you can ask your question next. Okay.
Hi, thanks for your talk. I have a question about the BME and how far we can actually apply it, because the initial SAC simulations that you showed, the simulations was actually good, quite close to the experiment. So is there kind of a limit that I can think of, like, okay, this is how far you can actually push the method?
Yes, there clearly is a limit. And it depends a little bit on the goal of what you'll do. And we've explored it, and other people have explored it in different ways. There's no, there's no simple, completely clear rule of thumb. But this phi parameter that I mentioned, which is easy to interpret, and this back to the previous question, it's easy to interpret when you have an unbiased simulation, because then it really measures the fraction of your initial trajectory that's left after reweighting. And if that gets much lower than, let's say, 10%, 20%, we see it be quite, then it gets quite noisy, right? And so what you can think about that is, essentially, minus RT, the logarithm of that is sort of a reasonable measure of the error in your initial ensemble, right? That is, how much do you need to push your ensemble? So if it's much smaller than that, you can still fit the data, and it in some sense gets better because you're fitting the data better. But of course, there's no intrinsic guarantee that it gets closer to the to the truth that we don't know. It's still, I think, a self-consistent approach, but it does get very noisy. I think one thing that is actually quite I think in hindsight quite nice is that this gets very clear when you're doing it. So effectively, if your simulation is very different from your experiment, you can see that it's difficult to fit it. Other methods, and we've definitely used those over years, where you bias the simulations, you can still fit the data also, but then the way that you do it, you effectively need to put in a lot of bias during the simulation. And so you still have the same problem. There's very little overlap between the distribution of your force field and the distribution that gives rise to the experiment. But you don't really notice it so much, right? This bias just goes in during the simulation. You effectively apply large biases during the simulation. And so it's the same kind of problem. It's just a little bit easier to observe here. What we've done in a couple of cases is we've sort of taken force fields that are increasingly good or increasingly poor and tried to reweight them against the same data. And we can sort of see them progressively, that this progressively just becomes, noise. And this this is where I get this number for sort of 10-20%. But, we've definitely done it with less overlap also. It depends also on, on the quality of the data and the quality of the sampling for these things. But yes, I agree. If your initial simulations are in very poor agreement with the data, this is not going to solve all your problems.
Thanks.
Okay, so David and Zach, you can ask yours next. David.
Sure, thank you. Yeah, thanks for the talk. My question was, do you have any ideas on how to best model a protein that has both structured and disordered regions, either in coarse grained or for all that?
Yeah, so in the Martini simulation, so some of the systems we looked at are like this. So in particular, actually both the tier 1 and the HNI-PA1, but in particular HNI-PA1 has, to begin with, 2 folded domains and a long disorder thing. Actually, this is the same A1 domain that we looked at the last bit. And I think we are safe there a little bit by this. So I mentioned that there looks to be some difference between how we need to rescale folded domains and disordered domains. It's not a huge difference. And so normally I would just say pick something in the middle, but actually the A1 low complexity domain is actually quite hydrophobic and compact. So even that domain actually, if we look at it alone, actually we don't need to change it so much. So that actually behaves a little bit like the folded domains. So there we rescaled it by 1.06-ish, and that works pretty well. So what we do is that we set up the system in Martini, we apply harmonic networks on the folded domains like we normally do in Martini. We delete all the context between the domains. We delete all the context from the the disordered bits and the link has. So there's no secondary structure, and that Martini cannot do, of course. And then that in the few systems we've looked at looks to be working really well. What we would really like is to have more systems like this. There are a few out there, but not very many. So really to try to figure out how well this works. But definitely this works pretty well. What's also nice that I didn't talk about, but I'll just mention it here, is that what we actually did in this paper is that Eric and Tanya did really nice salt-dependent measurements on the compaction of this system. And we actually capture this with the Martini. So we do this optimization of this rescaling at a single salt concentration. And then what they see in the SAX data and what we can also see in the simulations is that actually the interaction between the disordered bit and the folded domains is actually salt dependent. We can sort of titrate them off in the simulation. So we actually capture the salt dependency seen in experiments by the simulation. So this again suggests that the model actually captures something real about the interaction. There's also some NMR data that we can sort of compare with it. So it's it's not perfect, but it's definitely better than doing nothing, sort of picking something in the middle there.
Okay, Zach.
Hey, nice talk. I really enjoyed it. Okay, so I'm kind of interested in systems where we have less experimental data. So is there a way to kind of use this feedback in the other direction and kind of design experiments where we have low confidence in our simulations, and kind of like what would that look like?
Yeah, we've done a little bit of that, not too much, but we have sort of tried to try to have cases where we would sort of have different possible solutions, and then we say what are the kinds of data that would best. So we calculate a bunch of hypothetical data. This would be, for example, NMR data. We have two systems, two states. We have two solutions, and we say which NMR data would be most appropriate for distinguishing these two different systems, and then go in and then look. In this case, the NMR data existed, but go in and look at in more detail. So one can definitely do that. The other thing that we've done, which I think is something that we as a field should be doing more of, is again ask questions up front. Is it worth doing the experiment? Again, if you have possible different solutions, ask, say, construct ensembles that represent the different solutions, and say, how good would the data need to be to distinguish these different effects, right? So how small would the error bars need to be? So instead of saying, well, we have two different ensembles, say, well, if you have these two different ensembles and you did, let's say, X-ray scattering data, would you actually be able to tell them apart? Or so, for example, in this tier 1 system, I didn't talk about this. One of the nice things about, so it has 3 domains, and there's some really nice neutron scattering data where they actually made 2 of the domains isolated from the other domain. So one of them can be deuterated and the other one are non-deuterated, and then you can do native ligation, and you can essentially match out with partial deuteration the scattering of the different domains in a neutron scattering experiment. So you can essentially make one domain invisible, and so you could do a neutron scattering where you see just two domains in the context of the other one. And that's a complicated experiment to do. And so what we did in this paper, this was sort of after the fact, we sort of asked the question, well, how different would the ensembles need to be before you could actually, use this experiment to tell, for example, differences between, the distances between the different domains. And so I think that there's a lot of interesting things to be done in that area where we have ideas about what possible solutions are. We go to the sort of hypothetical experimental data and say, can we actually tell them apart before we try to convince someone to do the data? It's not very formalized, but I think that there's a lot of potential for doing this for also for EPR data and FRET data and all these other things. And another thing that I think is important to mention is, and this is something I think is also useful to think about, is that it's not always the obvious, so the best experiment is not always the one that is most sensitive to the changes that are going on. It's also something that you actually can plug into your simulations. So this is something that's particularly, we do a lot of work a lot with NMR data, NMR chemical shifts. The standard NMR experiment that people would look at would, for example, be the nitrogen proton HSQC, which is extremely sensitive to a lot of things, but it's almost impossible to calculate from simulation. So there's really no way of using it in simulations. So it's also important to think about experimental quantities that you're able to calculate from your structures, because otherwise you can't do this kind of merger. And so think about quantities that you can calculate from your simulations, but that you at the same time can measure relatively conveniently.
Okay, thank you so much. Let me actually thank you, Kresten, on behalf of everyone. So it will be really enjoyed. So you can tell from the stimulating conversation that we have had, we could speak actually 2 more hours about many other questions. But let me thank you. So I know it's 6:15 your time, as we are slowly getting into your dinner time. So thank you for joining us. And I also thank everyone to join us early morning on this side of Atlantic. So we are done with the seminar, so you can log out.